O-Level Biology 5090, Chapter 18, Biotechnology and Genetic Modification, Part 2, Genetic Modification. So what is genetic modification? Genetic modification is also called as genetic engineering and genetic manipulation. So direct manipulation of an organism's genes using biotechnology. And simply, we can say that genetic engineering is a process that uses laboratory-based technologies to alter the DNA makeup of an organism. So the DNA can be modified in this technique. Next is the recombinant DNA. So the recombinant DNA is a technology scientists developed that made it possible to insert a human gene. Human gene can be inserted into the genetic material of a common bacterium. So bacterium will act as a vector which will carry these human genes. So when this vector has these human genes inside, that at that time it is called as recombinant microorganism. And this recombinant microorganism would now produce the protein encoded by the human genes. In the next slide, we will discuss how the production of human insulin takes place by using these techniques. And students, you know, insulin is a hormone which is produced by the pancreas at the time when our glucose level is high. So insulin is secreted to control the or to balance the glucose level in the body. So there are some people who are diabetic, who are suffering with diabetes. So those persons are unable to produce the insulin, or insulin is produced in very small quantity. Why they produce or not produce the insulin? Because their pancreas cells are damaged due to some reason, or there can be some other reasons due to which they are not producing the insulin. So at that time, the insulin can be manufactured by this process, and it can be taken as an injection. So for this technology or for the production of human insulin, the scientists, first of all, they require a human being which has healthy genes. Healthy genes means the genes which produce the human insulin. So these healthy genes are taken from any healthy person, and then they can be inserted into the bacterium. So you know the bacteria has two types of DNA. One DNA which is in the cytoplasm without nucleus, and the other extra circular DNA which is called as plasmid. So this plasmid can be separated from the bacteria, and then it can be cut by using a special type of enzyme which is called as restriction endonuclease. So with the help of this enzyme, it can be cut, and then the genes which have gotten from the humans, healthy human, they can be inserted into this plasmid. So after inserting into the plasmid, they can be joined using DNA ligase. This is also an enzyme which will act as a glue. So this recombinant DNA will be now, it will be inserted again into the bacterium, where it will multiply. It will multiply, and there will be the production of more bacteriums with this recombinant DNA. Now they can be transferred into the fermenters to produce the insulin on large scale. So in the fermenters, they can be produced, and then from the fermenters, when the insulin is extracted, So at that time, it can be harvested for the use in the, you can say, medicine, as a medicine for different people who are unable to produce this insulin in the body. So in this way, the production of human insulin takes place. And the same techniques can be used in the production of different types of genetically modified plants. And here I would like to give one example, and this can be produced by the same technology, genetically modified organism, which will have insecticide resistant genes. So the insecticide resistant genes can be separated from the plant, and then bacterium, plasmid bacterium, and these genes will be inserted into the plasmids by using same enzymes, uh, restriction enzyme, and then they will be inserted into the plasmids, and these plasmids will be growing plant cells take up these insecticide genes from plasmid vectors. So the plants which are growing, they will get all these plasmids into them, and then the scientists can select them only those which have these recombinant or insecticide cells into the plasmid cell. Then those cells can be grown, and then they can change into the plants. And these plants which will be grown by this method, they will be resistant to the insecticides. So in this way, the genetically modified plants can be produced. And in the next slide, I would like to tell you the use of genetic modification in crop plants by inserting the genes. In the previous slide, I had told you how the insecticide resistant plants can be produced. So here I would like to say, first of all, in the first generation, it was started in 1994. The different types of plants were produced as herbicide tolerant, insect resistant plants, the virus resistance plants, the disease tolerance plants, the delayed ripening plants. So these were group of plants which were grown by these technologies. In the next generation, which was started in 1995, there was a batch of different plants which were grown by this technology, which were modified, you can say, by this method. The high nutritional value plants, which have different types of vitamins inside, or the extra quantity of vitamins in them, amino acid rich plants, premium quality, and low allergenic plants. So in this way, the different batch of plant was grown by using this genetic modification. And then in the third generation, which was in 1998, the plants which can have the qualities from which we can get the vaccination against the different diseases, antibodies, and pharmaceutical proteins. So in this way, the different other products were manufactured by using this genetic modification. That is, the vaccines were manufactured, and antibodies. We were able to make the antibodies, and we were able to make pharmaceutical proteins by this using techniques of genetic modifications. So genetic modification is helpful in the formation of different products. Now, when we use different technologies, there are some benefits of those, and there are some disadvantages or risk factors. So here we will discuss, or we will compare the benefits and risk of genetically modified organisms. So first of all, the benefit of genetically modified organisms, that is, the nutritional value of foods could be improved by using these techniques, genetically modifying the plants. We can introduce proteins, vitamins, or vaccines into them, it means we can increase their nutritional values. Whereas if we see the risk of this technique, then new traits could cause adverse health reactions. New proteins may cause allergic responses. So the some of the people, they can have allergy with the addition of these new nutrients into these plants. So the organisms, they can have different, not only the benefits, they can also suffer with different types of problems with them. The crops can be produced that lack new allergens. We can produce the crops or the plants which do not cause any allergy. So the removal of traits could have unknown effects, and sometimes the 50% risk factor is that we are growing the crops with new traits. We don't know any effect of those plants, so there is possibility that is it can affect or it can give benefit to us. The crops can grow in arid conditions for better yield by introducing drought resistant genes. So we can produce the plants which are drought resistant when there is no water, no nutrients to them, so they can grow. But crops may trade by diversity of local environment, increased competition with native species. But in other cases, you can say the crops which limit biodiversity. Biodiversity limit means there is less variations in them. So due to less variations, they have to suffer many problems. As for example, they need to compete with the native species which are already growing there. So in this way, they can have the different problems. So due to competition, whether they will grow or whether they will not grow. So it depends. Genetically modified crops can produce herbicides to kill pests. We can produce herbicide resistant plants. So the cross pollination could lead to super weeds. Cross pollination is super weeds which are harmful for the you can say plants. They depend on plants, they get nutrients from the plant, they get water from the plants, and these are the features of the weeds. But if there are super weeds, they are more harmful than weeds. So improve food supply, agriculture in poor countries. So we can increase you can say we can increase the production by this method. So by increasing the production, we can supply this to the countries which are very poor, which have very poor agriculture. They are unable to grow these different crops. So patent restrict farmers from assessing genetically modified seeds. So sometimes the seeds are not available to the people who want to grow these genetically modified seeds. Genetically modified crops may have longer shelf lives. They are not damaged after some time. They cannot, because they are resistant to different types of microorganisms. So they have long shelf life, and foods with genetically components may not be labeled. Sometimes when we manufacture the different types of crops by this method, they are not labeled. So when there is no labeling, we cannot get what is present inside them, how they are modified. So reduces the economic costs and carbon footprint, less need for land clearing and pesticide use. And different governments may have conflicting regulatory standards concerning safe usage. So in this way, the genetically modified organisms are, if they have some benefits, on the other hand, some risks are also due to use of those genetically modified plants. So students, it was all about the genetic modification. Please subscribe my channel, and thanks for watching.